Right. So now that we have completed the parts about Streptococcus, we will move on to Staphylococcus. Streptococcus, we got to Streptococcus by seeing that something is gram positive. Once it was gram positive, we saw that it was in the shape of cocci. When it was in the shape of cocci, we saw that it was catalase negative. So we did the catalase test, and when it was negative, we said these species are Streptococcus. Now, the ones that were catalase positive, we said that these clusters of cocci are Staphylococcus. So once something is Staphylococcus, we know that they're clusters of cocci and they're basically catalase positive. Once they're catalase positive, we move on to the coagulase test in order to differentiate whether it's Staph aureus or not. And if it's not Staph aureus, we do the novobiosine sensitivity test in order to determine whether something is Staph saprophyticus or Staph epidermidis. Now, the coagulase test. Why? How does the coagulase test work? Now, the coagulase is basically an enzyme, and what the Staph aureus does, it's important to know that the Staph aureus has fibrin in it. Um, the Staph aureus makes coagulase, right? So it hides itself inside the fibrin, and that's how the abscesses of Staph aureus are formed. This bacterium builds the fibrin wall, and the immune cells cannot reach it, right? The result is an abscess formation, and that's how in your body the pus-filled boils caused by the Staph aureus happen because it surrounds itself with fibrin. So the coagulase test determines whether something is Staph aureus or not. After the coagulase test, we do a further test called the novobiosine susceptibility test. Novobiosine is basically an antibiotic, right? Now we need to separate whether something is Staph epidermidis or Staph saprophyticus. So we place the novobiosine on an antibiotic and we place that disc onto two dishes. One is plated with S. epidermidis and one is plated with S. saprophyticus. Now, you can see that the one, for example, there's this ring around this disc. This means there's a zone of inhibition. The clear ring basically means that this dish is sensitive to novobiosine. And no ring around this disc means that this disc is not sensitive, it's resistant to novobiosine. And we know that the sensitive one is basically S. epidermis. This it is sensitive and it has a large zone of inhibition around the novobiosine, but the saprophyticus is not, right? So epidermis is sensitive, it has a large zone around it, but the S. staph saprophyticus is resistant. It has no zone around it. And that's how we do the novobiosine susceptibility test here, right? Moving on to Staph aureus, we said it was coagulase positive. And now a little bit about something about the enzyme itself. Now, when we come to Staph aureus, if this image basically shows you have these gram-positive cocci, these cocci, right? They're catalase positive, they're coagulase positive, they're beta hemolytic. That's something you can understand for later on. By the way, they're beta hemolytic, but I'm moving on to the fact that they look like clusters in this microscopic image, and you can see all these clusters. It's very important to know that they have this protein A, which is a virulence factor. This protein A binds to the FC portion of IgG, right? Once it binds to the FC portion of IgG, it inhibits the complement activation and phagocytosis. This is very important. Okay, now first, what is the FC region? An IgG antibody looks like a Y. Can you see this Y? So the Fab region binds the antigen, right? The FC region is like a handle. It's recognized by macrophages, neutrophils, natural killer cells, and complement proteins. The FC region is the part that tells the immune system, come eat this bacterium. Now, in normal opsonization, supposedly the IgG binds a bacterium, and this when the IgG is binding a bacterium, what happens is this FC IgG Fab portion binds to the bacterium. The Fab part binds to the bacterium, and the FC, the sticking outward FC fragment, this is sticking outside, right? This outward FC fragment can be recognized by FC receptors on neutrophils, right? So the antibody-coated bacterium attaches to the neutrophil, phagocytosis happens, and the bacterium is killed. But what happens with protein A is this protein A basically, instead of the antibody attaching normally, this protein A binds the FC portion of IgG. Since it binds this FC portion, this binding of the antibody on the FC portion, what this does is the FC bacterium is put in inside and the Fab is facing outwards. And what this basically does, um, this antibody is now attached backwards and it prevents phagocytosis. The neutrophils recognize these FC regions normally. So the bacterium has this IgG exposed and FC is exposed, but the Fab is not, right? And this evades the neutrophil receptor, right? What you would basically done by attaching this uh, FC portion, this FC fragment here is what you've basically done is you've bound the FC. The FC portion is now bound. Since it's bound, the Fab is exposed, but FC is not exposed. So neutrophil FC receptors cannot bind. Therefore, there's no efficient opsonization, there's poor phagocytosis, and the bacteria basically survives longer. I hope I was able to clear that. I took a minute to explain it because this part is important. So because of this FcIgG portion attachment, it inhibits the complement activation and phagocytosis. Now, it commonly colonizes the nares, ears, axilla, and groin. Causes include inflammatory disease, toxin-mediated disease. Now, diseases of Staph aureus, there are two kinds of diseases, right? One is the invasive disease where the bacteria itself enters the tissues, and the second one is the toxin-mediated disease. When we talk about the invasive disease, we're seeing that they have skin infections like furuncles, which are basically boils, and carbuncles, and osteomyelitis, where it happens in children more often. Then there's septic arthritis, there's endocarditis, there's pneumonia. So all of these are basically invasive diseases, right? This is where the bacteria itself is entering the tissue. Pneumonia classically happens after influenza. That's something to remember. Now we move on to the toxin-mediated diseases, right? When we talk about these toxin-mediated diseases, we know that the first one is the toxic shock syndrome. This toxic shock syndrome is basically a super antigen. Now, a normal antigen activates a few T cells, but this super antigen activates all the T cells of the body, right? The result is a massive cytokine storm. This massive cytokine storm, basically, what does it do? This massive cytokine storm gives you fever, hypotension, shock, a diffuse rash, and discommission. You get this scalded skin syndrome along with it because of the exfoliation of skin, and it's associated with tampons and nasal packing left on for too long. The exfoliative toxin, another toxin called the exfoliative toxin, causes the scalded skin syndrome. This is when the skin feels like a burn injury. And then another toxin called the enterotoxin. It comes from the word entero, so you know there's something related to the gut. This causes food poisoning. It's very important because it's heat stable. So cooking may kill the bacteria, but the toxin remains, right? So classically, you will have like a vignette where you will have like a potato salad or a cream pastry or mayonnaise. And 2 to 6 hours later, you will have vomiting diarrhea because the toxin already exists in the food, right? And then we have the methicillin-resistant Staph aureus, and this is due to this special gene called the MECA gene. This MECA gene is basically produces altered penicillin binding proteins. Since the penicillin binding proteins are altered because of the MECA gene, this penicillin resistance, beta lactams cannot bind. So you have to give special antibiotics here. And that is something you should know for this. Um, Now we move on to the other staph species. Now the other species, we have the staph saprophyticus and we have the staph epidermidis. We saw that novobiocin sensitive was the staph epidermidis and novobiocin resistance was the staph saprophyticus. Now staph saprophyticus is basically, we've seen it's catalase positive, it's coagulase negative, it's urease positive also, and it's novobiocin resistant. So it's important to know here that it's urease positive. Where it's found, it's basically normal, found, it's as part of the normal female genital flora in the perineum, right? Since it's found in the normal female genital flora, it's very common to get UTIs with this, right? It's the second most common cause of uncomplicated UTI. First is the E. coli, and the second is the saprophyticus, uncomplicated UTI. So always remember this. The first common most cause is the E. coli, and the second is the Staph saprophyticus. A typical vignette will be a young sexually active woman who has dysuria, frequency, gram-positive cocci, and it's novobiocin resistance. Once you know it's novobiocin resistance, you suddenly, you step by step know it's Staphylococcus. Now we move on to the staph epidermidis. For staph epidermidis, you must know that first thing is catalase positive, second thing is coagulase negative, third thing, it's novobiocin sensitive. It's part of the normal skin flora. Why is it dangerous? Because it makes this special biofilm. This biofilm, think about this like a bacterial glue, right? So organisms attach to plastic and metal because of this biofilm. So therefore, it infects all the things that are basically foreign things, right? Because it makes this film on the prosthetic valves, on the hip implants, on knee replacements, on IV catheters and central lines, right? So it contaminates all these implants, right? That's something to know about epidermidis. And I'll pause the video here and we'll move on to our next segment.